Welcome, everybody, to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Emilio Mendez Olivos. I'm a postdoc at University of Calgary, and I will be moderating today along with Dr. Lauren Cote, a postdoc at Stanford University. We are super excited to showcase the work of our standing postdoctoral members. And for today, we will have Dr. Jennyfer Mitchell from University of Colorado and Wade Sugden from Boston Children's Hospital Medical School to share their research. Each speaker will give a 20-minute talk followed by a 10-minute Q&A. So please enter all your questions at the Zoom Q&A box, and we'll be reading them. Now, I will like to introduce Dr. Jennyfer Mitchell. She earned her bachelor's degree in biology and a master in marine biology at the Universidad Nacional de Colombia in Bogota. Her initial research focused on studying sponges, analyzing extract for antibiotic properties and correlating bacterial diversity with susceptibility to colonization. This work contributed to the development of anti-fouling additives. For her PhD, she continued working with peripheral phylum in the United States now at the University of Denver, alongside Dr. Scott Nichols. Together, they describe how adherent junction and focal adhesion proteins are often co-distributed in these animals, in contrast to the segregation that is seen in all invertebrates. In 2018, Dr. Mitchell completed her PhD and developed a passion for zebrafish, as many of us. She then moved to the University of Colorado, now joining Dr. James Nichols' lab to study frontonasal development. In 2020, her project was funded for 4 years, thanks to the Ruth Kitchener Postdoctoral National Research Service Award. And throughout her career, she has been recognized with nearly 10 to 20 awards, including Postdoctoral Poster Award during the Annual Dental Research, Presidential Membership Award from the Genetic Society of America, from the same society, the Attendance Support Minoritized Scientists, Rising Scientist Award, and very recently, she received the Best Postdoc Poster Presentation at the Southwest Regional Meeting of our beloved society. She has contributed with 12 conference posters, 8 conference talks, 4 invited seminars, actively involved as a TA, tutor at every institution. She has been first author of 8 out of 11 publications, including a book chapter. Her outreach activities are huge, and she's currently in the job market. So please welcome Dr. Mitchell and her presentation today.
Thank you so much, Emilio, for such a lovely introduction. I'm going to share my screen now. I also would like, in the meantime, to thank all the organizers of this seminar for such a great opportunity to present my work today. Okay. So let me start by introducing my model system. I work with zebrafish, and what you're seeing here is the most ventral view of a zebrafish larva, in which progenitors are labeled in magenta, and progenitors that are turning on chondrification programs. Those progenitors that are turning into chondrocytes are labeled in green. And I'm mainly interested in 2 structures here in this time lapse, the trabecula and the ethmoid plate. These are 2 structures that build the most, that are simplified in the zebrafish, and they are the mid-facial skeletal structures in these early stages. Now, one question that we can answer is, how did this animal knows where to build these structures? This type of question has puzzled developmental biologists for years. And with the discovery of the Hox code, the Hox genes, it revolutionized how we address this type of questions. So now we know that the animal body plan is built of segments that conserve homology among them. And then each of these segments has a specific genetic address that is going to give a specific identity to each of these segments, and it's also going to define what organs are derived from each of these segments. Now, when these genetic addresses derail, we get things like this. This is a posterior to anterior formation. It's a classical example that comes from Drosophila, in which the antenna got transformed into a leg. So these tell us 2 things. One, genetic addresses can be moved. So we can take one genetic address from one portion of the body and insert it in a different portion of the body to change what are the organs derived from it. And second, this is possible because of that homologous nature of the segments that build the animal body plan. And this is a beautiful example that comes from invertebrates. But we're also interested in understanding how the skeletal, how the skeleton of the head is built in vertebrates. So in my first publication in the Nichols lab, we find out that neurocrine cells are divided in these specific clusters. Now, some of those clusters that we identify transcriptomically reflect things that we already know about the embryo. This is a 24-hour zebrafish embryo where I'm showing you the most known parts or segments that build the head, like the anterior arches, posterior arches. In our survey, we also identified a subpopulation of neurocrine cells that we label frontonizal. You can see it right here. No matter how we cluster these cells, there was always going to be a subcluster that it was going to be separate from all the other cranial neurocrine cells. So we start to wonder what is setting up the identity for this frontonizal neurocrine cells. But to answer that question, we first need to understand how the rest of the head is organized. So I have mentioned that cranial neurocrine cells in the embryo are organized in these homologous segments that we call pharyngeal arches. And each of these pharyngeal arches identity is set by homeobox containing gene families. So we know, for example, that the DLS code determines different domains in the pharyngeal arches 1 and 2 to give rise to all the structures that I have color-coded down here in this representation of the side view of the zebrafish larva. We also know that the host code, for example, is important for anterior-posterior determination in these arches 2 to 7. But then we were wondering what happened in the gray area. So I have depicted some skeletal elements in gray because we don't have a clear understanding of what is setting up the pattern of these elements. And those are elements of the midface, as I showed you in the first movie. And so we're wondering, is it possible that this homology in between segments is conserved across the whole head? Examples of homology or conservation of the homology among the pharyngeal arches come from examples from this one, in which mouse mutants, mouse is an enzyme required to maintain the host code, and upon removal of that, we derail the host code, and we can see that elements of the arch pharyngeal arch 2 disappear, and instead we have a duplication or a mirror image of elements of pharyngeal arch 1. So we have here the PQ and the Meckel's cartilage duplicated. So is it possible that these homologies conserve throughout the whole full head? To answer that question, we start to look in detail at these skeletal elements that we're not clear about the patterning and what sets their identity. So let me walk you through these specific elements. So this portion here in gray, I have been dissected out and then put in a flat mount. And we can see here in Alcian blue is the cartilage and the alizarin red for the bone. We can see that the ethmoid plate is right here in the front, is divided in 2 zones, the lateral and the medial. We have paratrabecula and 1 parasphenoid bone that build those structures right there. So we have this frontonizal neurocrine cell population that is segregating from everything else. And then we have some skeletal elements whose patterning is not absolutely clear. Can we link these two concepts, these 2 elements together? To do that, we start to look at the landmark genes that are setting up the transcriptional identity of this population. And to no surprise, we find that there is a homeobus containing gene family right at the top of the landmark genes in this frontonizal neurocrine cell population. The ALS genes have been around for a while. We know them very well because they are linked or associated with frontonizal dysplasia. This is a set of phenotypes that appeared naturally in humans that have mutations for this gene. And the defects are focused right there in the middle, in the mid-facial portion of the skull. Now, this is in humans, but now we want to understand how these genes are being expressed, what is their function. So that's why we go back to the zebrafish. And here I'm showing you HCR in situ hybridization images in which we corroborated the predictions from our transcriptomics. We can see that ALX3 is the gene that is most broadly expressed in the most frontal portion of the embryo. It is around the nasal placoid, in front of the eye, a little bit dorsal to the nasal placoid, right in front of the eye, and it stops right there at the chorioid fissure. So the chorioid fissure is a specific landmark of eye morphogenesis, and it's going to be very important for the set limits of where ALX expression happens. Now we can see that ALX4A has a more restricted expression pattern. It's more towards the ventral portion of the nasal placoid, and also we have expression in eye cells. Is the only one. And then we have ALX1, is the one that has the most restrictive expression at this stage in zebrafish. So it's right there in between the nasal placoid and the eye. It's still a little bit in front, anterior to the eye, and it stops right there at the chorioid fissure. So you can see that all of them are expressed right up to that specific landmark in the eye. And then something I want to get your attention is that all three genes are expressed right there in between the nasal placoid and the eye, what we can call like the bull's eye, because around that area, we're going to have expression of two ALX members, or maybe just one ALX member. So it has a little summary of this expression analysis. We can say, well, we have a concentric or nested expression of ALX genes. And these let us, just like it happens in DLX, these led us to the hypothesis, well, maybe we're seeing an ALX code, or this nested ALX expression is regulating the frontonasal neurocrest identity. To start answering that question, we start to study ALX function. We created different alleles for each of these ALX genes. And in our first screens, we only saw robust craniofacial phenotype in ALX3. So let me walk you first through this diagram in the wild type. So you can see here that the ethmoid plate is divided in two zones, the lateral and the medial. The lateral has these cells that are more columnar. They stain darker in Gaussian blue because this is a readout of how much extracellular matrix deposition has been happening, versus the medial cells are more round, less extracellular matrix is being deposited. So we can tell those two zones perfectly fine there. But when we remove ALX3 function, we can see that that cell morphology disappears. We don't have these medial versus lateral domains anymore. We also have ectopic cartilage at the posterior portion of the ethmoid plate, and then reduction of the parasphenoid point. But I want to get your attention to this phenotype, this cell morphology. To understand that phenotype, we put this allele in the context of the SOX9 GFP to label again chondrocytes as they are being differentiated. We can see in the wild type there are two clear zones where the lateral chondrocytes are the ones that are coming up first in development. And you see the medial portion is pretty much devoid of chondrocytes. But then that's telling us that these come out later in development. But then when we remove ALX function, all chondrocytes are coming undifferentiated, are coming up in one single front. It's almost like we have a lateralization of the ethmoid plate. So we can conclude two things here. ALX3 is important to determine a differentiation timing in chondrocytes, but also it hints us to the idea that, well, maybe we're losing a cell type, maybe we're losing cellular identity. And then to continue testing this hypothesis, I start to put together all the other alleles that didn't give me a phenotype in the context of ALX3 mutants. And again, this is a wild type image of that beautiful ethmoid plate. And then when we remove 2 copies of ALX3, we see a more dramatic phenotype. It's almost like the medial portion of the ethmoid plate is gone. And instead, you can see this area here is a little bit blurry. That's because these are flat mounts that I squeezed together. So the cells, these are actually ectopic chondrocytes that are sitting up there on top of the ethmoid plate, sorry. And then when we squeeze them, it looks like that. But when we have the whole mount of the animal in lateral view, we can see that there is ectopic cartilage right there on top of the ethmoid plate. Now you can also see that the oral face of the oral shape of the head is different, is more flat. And this is something very interesting because in humans that carry these similar mutations in the ALX3 gene also have that appearance of flat face. And a little treat of my some of my future experiments is this adult fish, in which you can clearly see the flattening of that face that is happening through, all the way to adulthood in these mutants. So to understand this phenotype, we performed two experiments. The first one is a lineage tracing with the Sugden Cote line, and I'm leaving here my in situ hybridization so you can see where all ALX members are expressed right there in between the nasal placard and the eye. This is very important because it was help us to determine what cells we wanted to photoconvert during the Sugden Cote lineage tracing. And you can see here, we photoconverted those cells in between the nasal placard and the eye. This is a zoom of that area right there. And then you can see that 4 days post-fertilization in the very same animal, we're able to see those cells sitting right there in the same position in between the nasal placard and the eye and on top of the ethmoid plate. So this is also an enlargement of that little square right there. So this is telling us these cells are sitting right on top of the ethmoid plate in between the nasal placard and the eye. And then when we put this SOX9 FGFP transgenic line in our mutant context, in this case, we're also looking at ALX3, ALX4A double mutants, we can see that those cells that were sitting right on top of the ethmoid plate are also turning on the chondrification programs. So cells that shouldn't be chondrocytes are turning on the chondrification program. Now, this is very telling, but we really wanted to make sure that we're talking about a change of identity. And the best way that we have now to prove this is with transcriptomics. And that's what we did. So we did single cell RNA sequencing analysis in wild type, ALX3 singles and ALX3 doubles. But I'm going to focus right now on the doubles for the sake of time. And then you can see here in this UMAP that the frontonasal clusters are divided now in two. This is frontonasal 1, frontonasal 2. And then we have arches right here in the middle of that UMAP. So this is the center of that UMAP is the dorsal and ventral arches or cells that segregate with the dorsal and ventral identity, and then intermediate right here. But I want to get your attention to this portion of the UMAP right there, where it's almost nothing. But then when we lose two copies of ALX, when we remove the function of ALX3 and ALX4A in our mutants, it's almost like we have this ectopic cluster, just like we have ectopic cartilage above the ethmoid plate, we have an ectopic cluster in this UMAP. And this is corresponds with a loss of cells from the frontonasal cluster 2. Now, just by looking at the cell counts, we can tell that we are gaining pharyngeal identity at the expense of frontonasal identity. So we're seeing changes in the transcriptomic signature of these cells. Now, up until here, I have shown you results about single ALX3 mutants and the doubles, ALX3 and ALX4A, but there is one more member, ALX1, and ALX1 is the gene that has the most dramatic phenotype in humans. So what happens when we put together all these alleles? So let me walk you again a little bit through this dissection because it's a little bit different. So besides the ethmoid plate, the trabecula, and the parasphenoid bone, I have also included the first ventral element, which is the palatoquadrate and the pterygoid process that articulate with that ethmoid plate. And when we remove all 3 copies of ALX in the zebrafish embryos, what we have is pretty much I cannot distinguish ethmoid plate. We can barely distinguish trabecula or bone. Instead, what we have is like miniature versions of structures that look like that ventral element, the palatoquadrate and pterygoid process. This one, for example, is the one that looks like a little miniature version of this. And this is just one mutant right there. And here's another example in which the bone was better stained. And we can see that that bone, well, doesn't really look like parasphenoid anymore. Instead, it's kind of like lining that cartilage, just like the endothelial bone is doing it for the palatoquadrate and pterygoid process. So all this is pointing out that we have a skeletal transformation. In this case, from the midface is being transformed in the very first ventral skeletal element, which is the pterygoid process and palatoquadrate, an upper jaw element. Now, doing single-cell transcriptomics in triples is logistically challenging. So what I'm doing to study or to confirm that this is actually natural transformation is looking at genes that are very well known as markers of pharyngeal arches, like DLX2A. And you can see here in this UMAP that his expression pattern is completely different from ALX3. And here you can see in the wild-type embryo, is perfectly lighting up in those pharyngeal arches, and then in the forebrain and a little bit in the hindbrain. Upon removal of ALX function, we can see the ectopic expression right there in the frontonasal area. So we can imagine here is the nasal placoat, and right here is the lining of the eye. So right in front of that area where I show you all the ALX members are expressed. And this happens in the triples, and this happens also in ALX3 and ALX1 and ALX3 and 4A down mutants. So with this, I can summarize a little bit here, and I can tell you we have found a cranial neurocrest cell subpopulation that segregates from all the other subpopulations, whose identity is determined by a homeobose containing gene family, just like it happens in all the other arches, whose expression is concentric, overlapping, just like it happens with the DLXs. And when we remove the function of this family, we're having skeletal transformations, in this case from the midface to the upper jaw. Now, this has repercussions both in our understanding of the disease of frontonasal dysplasia. So maybe what we're seeing in these patients is possible structural transformations. And then this is an also very exciting result that has evolutionary implications, because now we can tell that maybe there is more, there is an anterior segment to pharyngeal arch 1. And this is what is known as the arch 0 hypothesis. Back in 1937, De Beer proposed that there was an ancestral homologous pharyngeal arch residing anterior to arch 1, that it was responsible for the most rostral portion of our face. Now, in the 2000s, the PI of my PI and good collaborators, estimated collaborators that we have, they also did lineage tracing, they did some gene expression studies, and they determined that the pharyngeal arch 1, especially the maxillary domain, is the one that is give rise to these frontonasal, to these midfacial elements.
1 minute, Jennyfer.
Okay, thank you. I'm almost wrapping up. And they confirmed that this arch 0 hypothesis was probably something that was a little bit too ambitious to propose by De Beer. However, under the light of ALX function and how is this family that is setting up the identity of this the specific subgroup of cranial neurocrest population, we need to revisit this idea and maybe further look into this arch 0 hypothesis proposed in the early 1900s. And with this, I would like to thank everybody that has directly contributed to the experimental portion of this project. But I would also like to thank everybody that has been supported throughout all the years in my formation as a scientist. I would love to name you all, but I have put you here in this slide, and it's with a heartfelt that I really thank you for all your support through all these years of postdoctoral training. I also like to thank our funding sources and all the societies and local programs at CU Anschutz that had supported me throughout the years as well. And with that, thank you all for attending today, and I'm ready to take your questions.
Thank you, Jennyfer. We have already burning questions in the chat that I'm going to read for you. Mm-hmm. Sally Moody asked, what are the human mutations? Are they knockouts like your fish or single nucleotide amino acid / amino acid changes?
They are point mutations.
Point mutation, perfect.
Mm-hmm. Yes.
Sally then asked, are there olfactory nerve defects in the ALX mutants since the ethmoid plate is perturbed?
That will be something that we would love to see. I'm pretty sure that these ALX positive cells, as you saw it, they are mostly in the soft tissue. So I'm pretty sure there is going to be some sort of defect that we're going to find in that soft tissue too. I mean, we're seeing the skeletal readout in this project, but certainly I feel, is my gut feeling, that there is more to understand about ALX function in some other tissues, whether it's direct or indirect. That's something that we need to determine in future studies too.
Okay. Grant and Jindal, really nice talk. Other than the ALX genes, are there other Hox genes expressed in the frontonasal region?
Well, actually, and that's something I probably skip a little bit on my talk. This frontonasal cluster is Hox negative. So there is no Hox genes. There are some other homeobus containing gene families, like PITX, and I can't remember what is the other one, but MXX is also expressed there. But no Hox genes are expressed in the frontonasal cluster.
Okay. Gage Cramp asked, the DLX2A expansion is really impressive. Do you also now see distinct zones of HAN2, DLX3, 4, 5, 6, PO3, F3A, etc. in the transform ARC0 domain of ALX mutants?
Those are experiments that are waiting for me at home. I definitely want to look at other genes, and I'm going to, I want to be a little bit more systematic. Now that we have this nice transcriptomic data set in between wild type and mutants, I want to go to those genes that are like really dysregulated in between those two samples, and then start to do in situs to confirm that expansion too. But definitely we can also add in genes like what you're suggesting. Yeah.
Perfect. Munisa Junaid asked, how is the oral ectoderm affected in the ALX mutants? Also, are the ALX genes interchangeable with one another? If one is gone, the other takes place, some sort of redundancy function?
Yeah, well, I haven't seen expression specifically in the oral ectoderm, expression, ALX expression in the oral ectoderm. It seems like it's just very well restricted to that mesenchyme, the frontonasal mesenchyme.
Okay, and is it affected also?
You know, that's a good question, because I haven't like particularly looked at the changes in the oral ectoderm. So that's a very good question, Minisa. I think we should probably look into that too. And then to answer your second question, I believe that there is, I mean, it's the nature of working with paradox in zebrafish, right? There is going to be a little bit of redundancy. So I already did some experiments in which when I knocked down ALX3, the domains of expression of ALX4A and ALX1 change a little bit. The overall expression doesn't change that much, but the domains like the overlapping. I measured the overlapping in between them, and that changes a little bit when in my ALX3 mutants. I will have to see if this is also happening in the doubles. And as I believe that there is some redundancy in their function, I also think that there are specific functions that each of them are carrying. And that comes not only from observations in zebrafish, but also the human phenotypes. I mean, we can see each of these individuals that I show you in this slide, they have just one point mutation in one specific ALS. And that's why we have a classification of frontonasal dysplasia 1, 2, and 3, because they're distinguishable in between them. So I believe that there is also some function specific to each of these ALX genes.
Okay, one final and quick question, and I need a quick answer. Any plans, Tim Nguyen asked, any plans for overexpression paradigm? This will set up a prediction that you lose the RNarch, for example, loss of DLX2A.
Wait, can you say that question again? You cut off a little bit.
Any plans for an overexpression paradigm?
Oh, yes, yes, of course, because we don't see ALX expression starting like really, like the maxillary domain has very little expression of ALX. The maxillary domain of ARCH1 has very little expression of ALX. So I think we definitely will want to overexpress ALX in other pharyngeal arches and see what programs are being affected and what skeletal outcome of that is going to be. That's, yeah, that would be a beautiful experiment to do.
Perfect. Thank you so much, Jennifer. Lauren, it's all yours.
All right, thank you so much to Jennifer and to my co-moderator, Emilio, for a really wonderful talk, Jennifer, and a great discussion. My name is Lauren Cote. I'm a postdoc in Jessica Feldman's lab, and it is truly my pleasure today to introduce Dr. Wade Sugden. He's currently an instructor in pediatrics at the Harvard Medical School in Tristan North's lab. There, he's studying how fluid flow impacts transcription during blood development. Wade is a longtime member of the zebrafish community, and he got his start in zebrafish when he was an undergraduate at the University of Oregon in Charles Kimmel's lab, studying craniofacial development, and got a bachelor's degree in biology there. He then moved to Europe in Germany, where he had a very productive graduate career with Arne Siekman at the Max Planck Institute for Molecular Bioscience in Munster. There, he really beautifully investigated how biophysical forces of blood flow help shape and pattern the zebrafish vasculature. He then turned from the vasculature to studying the blood stem cells and moved to Tristan North's lab, where he is now studying how biophysical forces of blood flow actually control hematopoietic stem cell development via the transcription factor gap. And there he's using both zebrafish and human iPSC models. All of this work has been recognized with 2 really generous NIH awards, an F32 postdoctoral fellowship, and a KO1 Scientist Development Award. In addition to his really gorgeous research on blood development, he's been an active leader of both his graduate and postdoctoral communities, has mentored multiple students, and has taught several hands-on courses. Wade is currently seeking tenure-track opportunities to build upon his work on how mechanical forces are integrated into gene programs. And I'm really looking forward today to hearing his talk entitled Blood Flow Directs YAPTAZ-Mediated Transcriptional Regulation of Self-Renewal Programs to Control Development of HSPC Expansion by Mechanical Stimulation of Piezo. Take it away.
All right. Well, thank you very much, Lauren, for that very kind introduction. And I really want to thank SDB and the organizers of this seminar series for inviting me to present my work today. It's a real privilege to be here. Like Lauren said, today I'm going to be telling you a bit about how mechanical forces from blood flow through this vessel, this is the dorsal aorta, in the trunk vasculature of a zebrafish embryo, is important for promoting the production of hematopoietic stem and progenitor cells during development. So I'll take a quick step back and say that I've been motivated by this question of how do cells create functional tissues for quite a while. On the right here is a picture of the first transgenic model organism that I ever made while I was an undergraduate in the Kimmel lab at the University of Oregon. And shout out to Chuck and some U of O alumni that I see are on the call today. So this is a zebrafish that expresses GFP in the craniofacial chondrocytes. And as developmental biologists, we can appreciate that one answer to this question of how cells make tissues is in their genetics. And Jennyfer gave a beautiful talk that really highlights this. So these are things like morphogens that help the cells get to the right place, the structural and motility machinery they need to undertake that task, and differentiation to become the right cell type. But in my PhD studying vascular biology, and now in my postdoc studying blood, I've gotten really interested in this idea that cells need a kind of plasticity and responsiveness to adapt to the function of the tissue that they're in as it begins to work. And so I'm going to show you a couple movies that highlight what I mean. This first one is of some of the first blood vessels that develop in a zebrafish embryo. And what I want you to appreciate is that it's not just that the endothelial cells have gotten here and built a blood vessel and then it's over, that the actual function of this tissue exposes them to a kind of continual dynamic mechanical cue that they need to be able to adapt to. And that's not only true just in embryonic development, it also applies in adults. And this is a regeneration setting, again in zebrafish without any particular labeling, where you can see red blood cells coursing through these newly developing blood vessels. And so I just want you to keep in mind this idea that there needs to be an interaction between genes in the cells and cues that they get from being in a functional tissue. And the vascular system is a great place to ask these kinds of questions because it's really a place where form meets function. And so this is just a textbook diagram for people that don't think about blood vessels all the time. You can see at the bottom, this is the hierarchical organization of the vascular tree. with large arteries that progress into very tiny capillaries that then collect blood into increasingly large caliber venules and veins. Above that are some of the hemodynamic properties of the blood flow in different parts of the tree: high pressure in the arteries, low pressure in the veins, high blood flow velocity in the big vessels, and very slow in the capillaries. And this relationship is not just by chance. In fact, it's built upon this ability to integrate a mechanical cue with genetic instructions. And I'm going to show you a movie that if that fails, you get total tissue pathology. So if you're a human or a zebrafish shown here with a mutation in the endoglin gene, you get arteriovenous malformations and the whole mechanical properties are off. You get blood flow going the wrong way through some vessels are bleeding, others are collapsed and have no flow. So I just want to make the point that this form and function integration is not just interesting for development, but it has real disease implications. So today we're talking about the blood. The blood is a pretty huge component of the human body, and most of half of the fluid going through your vessels is liquid plasma. The rest is cellular constituents. Most of those are red blood cells, and then the rest are immune cells. And a key feature of the system is a short lifespan and high turnover of all the cells. So that means you really rely on progenitors to keep producing blood cells daily throughout your life. And in the North Lab, we're studying hematopoiesis, which is the process by which the differentiated cells of the blood are formed from stem cells. So that's shown here in the right in this simplified hierarchy, where you have hematopoietic stem cells, or HSCs, at the top, which are able to self-renewal or differentiate to give these other more restricted progenitors, and ultimately the red blood cells and cells of the innate and adaptive immune systems. I'm going to leave it at that for today. During development, there are multiple waves of blood formation, and some of them come from developmentally restricted progenitors and not through this HSC intermediate. But it's true that embryos make these HSCs and they can persist into adulthood and contribute to all the blood. They're very rare, and that makes them extremely therapeutically valuable, because if you can harvest them successfully from a patient, for example, you can restore the complete blood system. And so for that reason, it would be very therapeutically beneficial to be able to expand this rare pool or make them de novo. And so we study where do HSCs come from in the embryo to try and see if we can find cues that might help achieve those goals. And so what's pretty interesting for a vascular biologist is that HSCs actually come from endothelial cells from a blood vessel in development. And in a mouse, this would happen in a region called the aortic gonad mesonephros, or AGM. And if you take a cross-section through that tissue and focus on the dorsal aorta, which is the largest artery in the middle, and examine the ventral wall, you would see that a subset of the endothelial cells here can acquire the expression of RUNX1, a master regulator transcription factor that can change their identity from endothelial to blood-forming stem cell, and they actually can go through a total change and detach from their neighbors and enter circulation as an HSC. And that process is called the endothelial to hematopoietic transition, or EHT. So it takes lots of transcription factors to make an HSC, not just RUNX1. And one take home that I want you to appreciate is that the transcriptional logic is highly recursive. So for example, you need this hematopoietic triad of TFs, FLE1, GATA2, and SCL to get RUNX1 expression. And then all of these TFs regulate not only themselves, but each other. And this network has only gotten more complex as technologies let us find more enhancers and add transcription factors to the network. I also want to say that EHT does not depend only on these cell intrinsic transcription factors, but on interaction with the environment as well. And we know this from our model systems for studying EHT. So it's possible to take human iPS cells in vitro, for instance, through directed differentiation that tries to recapitulate this developmental ontology. And you can get arterial endothelial cells, a hemogenic endothelial population, and they will actually undergo an EHT process and bud into the supernatant, expressing blood stem cell surface markers, but they don't function like true HSCs. So they're relatively short-lived and have limited multi-lineage potential. So for that reason, we come back to this great in vivo vertebrate model zebrafish, where the expression and function of RUNX1 is conserved. And I'm showing that here by in situ hybridization. You'll see lots of pictures like that today, where we can ask, what are key developmental cues present in the embryo to make HSCs that we might be missing in these in vitro models? So one of those critical developmental cues is actually the mechanical force from blood flow in the vessel. So I'm showing you here the dorsal aorta with a budding hemogenic endothelial cell. Our group and others were among the first to show that wall shear stress, which is a frictional force parallel to the direction of blood flow sliding across the endothelial layer, promotes a number of small molecule effectors like nitric oxide, prostaglandin, and calcium that's critical for making HSCs. When I started working on this question, nobody had investigated cyclic stretch in isolation, and that's an orthogonal force caused by an outward pushing of the blood on the vessel wall. And the bigger picture question of how do you actually go from either one of these forces to gene expression changes in hemogenic endothelium was not well understood. So a few years ago, we published a dorsal aorta on a chip microfluidic system that allowed us to culture human iPS-derived CD34 positive hemogenic endothelium under static conditions or subject to wall shear stress or cyclic stretch. We saw that both forces led to the upregulation of the RUNX1 transcription factor, but interestingly, only cyclic stretch led to the upregulation of target genes of the YAP transcription factor. This was true in human cells, and then we moved into zebrafish to show by loss and gain of function genetics that YAP was in fact required for developmental hematopoiesis. So what is YAP? YAP is pretty interesting if you're thinking about a way to go from a mechanical cue to gene expression changes. YAP and its paralog TAZ are transcriptional co-activators, and their subcellular localization is determined by their phosphorylation status. So canonically, that's deposited by these hippokinases, and if phosphorylated, YAP and TAZ are targeted for degradation. It's been known, though, for more than 10 years that mechanical force can directly relieve that inhibition and allow them to go to the nucleus to regulate their targets. This is the protein structure on the right. YAP and TAZ are both paralogs, so they're extremely similar. And the key take-home points here are that neither transcription factor bind the DNA directly. Their main DNA interacting protein is TEAD, but they can interface with other DNA binders by these WW domains. And that includes RUNX proteins. And given the importance of RUNX1 in hematopoiesis, that's going to become important later. So in the publication we showed that this Rho GTPase mechanotransduction cascade is conserved in hemogenic endothelium. But the open questions that I'm going to talk about with the data I show you now are what are the genes that YAP controls to regulate the hematopoietic program? What DNA binding partners are responsible for YAP-dependent hematopoiesis? And what actual components on the membrane help sense force and relay you to this YAP activity? So this will be the last piece of published data I show you. We think that YAP is required for maintenance, not initiation of the hematopoietic program in hemogenic endothelium. So on the left is a cartoon of EHT progression in zebrafish with different markers, and on the right is the data. What we see is that YAP homozygous mutants are able to specify a population of GATA2B positive hemogenic endothelial cells in their artery, but they really fail to expand RUNX1 and CMIB expression, and it just stays a salt and pepper distribution in the trunk. So that makes us really curious. What are the essential YAP-regulated genes in this population that help promote the hematopoietic program? So to do that, we did transcriptomic analysis on sorted endothelial cells from dissected tails. And we analyzed wild-type zebrafish, YAP overexpression animals, and YAP loss-of-function animals. And I'm showing you here, I just pulled out the clusters relevant to EHT, including the arterial endothelium, hematopoietic progenitors, and macrophages as a differentiated stage. If I show you the 3 transcription factors that were on the last slide, we can see that they're all highest expressed in the hematopoietic progenitor cluster, but GATA2B is unique in having a distribution that notably extends into the arterial cluster. And I'm showing you here a more binary yes or no for GATA2B. We think we can use this to define arterial hemogenic endothelial cells. I'm calling them AHECs here. If we do differential gene expression on these AHECs, we can see that YAP has context-specific effects here. And pathway analysis shows a nice overlap in pathways associated with genes that are up when YAP is overexpressed and down in the mutant, including cell cycle, metabolism of lipids and RNA, and right here, transcriptional regulation by RUNX1. So this really suggests that YAP can be augmenting the function of this master regulator hematopoietic transcription factor. And that's more clear here if we look at a curated list of hematopoietic transcription factors and differentiation genes. What we see is that these hematopoietic self-renewal TFs that I showed you earlier are generally up when YAP is overexpressed, lowered in expression in the mutant. And strikingly, some of these myeloerythroid differentiation genes are generally low when YAP is high and up in the mutant. So what we think is happening is YAP is helping to promote a self-renewal and stemness program in this hemogenic endothelium to promote EHT. So what does YAP work with in the nucleus to do this? To try and answer that question, we employed a heat shock dominant negative YAP model. And the way this works is it overexpresses an N-terminal piece of the YAP protein that can interact with all the DNA binding proteins, but doesn't have a transcription activation domain. To our surprise, when we did the experiment, we got a kind of gain of function phenotype where we saw more RUNX1 and c-MYB positive cells here by in situ. And to make a long story short, we did some really interesting experiments to try and track this down. It reveals a compensatory role for TAZ because this hematopoietic rebound we get in the dominant negative YAP animals doesn't happen if we do it on a TAZ mutant background. And you can show this quantitatively by FACS for transgenically labeled HSPCs. With a really tiny dose of morpholino, you can blunt that effect completely. So this is the first in vivo data we have that TAZ can play a role alongside YAP in hematopoiesis. What DNA binding proteins do they use to drive this effect? So to try and answer that question, we went in vitro using luciferase assays. This here is essentially a giant control experiment using a T-driven luciferase construct, which drives lots of expression if you use any number of YAP or TAZ variants, except this variant of YAP, which cannot interact with TEAD. Next, we used a luciferase construct driven by RUNX enhancers, and you can see that neither YAP or TAZ can drive expression from this construct. RUNX1 by itself has a modest induction, about 4-fold, and then there's a real synergy between RUNX1 and TAZ, not RUNX1 plus YAP, which more than doubles expression of this construct. And that synergy is very specific to this TAZ-RUNX interaction because you get it with TAZ wild type, a constitutively active TAZ, and you can lose that completely if you make point mutations on TAZ at this interacting domain. And so we're going back into the zebrafish with some of these variants where we think we can show nicely which ones of these are required for the hematopoietic effect. And so in the last couple of minutes, I'm just going to spend some time on this final question that we have, which is how is force at the membrane actually sensed and transduced to this YAP activity that I've described to you? And a candidate membrane protein that we're really interested in is piezo1. Piezo1 is a huge multimeric stretch-gated ion channel on the membrane. And the way it works is upon membrane deformation, there's a pore that opens right through the middle of the channel to allow cations into the cell. One reason we're so interested in this protein is there has been a connection between piezo1 and YAP signaling established now for quite some time. And we're fortunate in that there's a chemical agonist called YODA1 that's very specific for the piezo1 channel and not the related piezo2. And what I'm showing you here is that this is in HeLa cells. This is shown in other cell types as well, that stimulation with YODA1 will increase YAP nuclear localization. And so we wondered, might this be conserved in hemogenic endothelium in zebrafish? So first, we treated embryos with this chemical agonist for piezo1. And we see that, in fact, the YAP target gene CTGFA is increased by qPCR. And we also see an increase in RUNX1 and c-MYB positive HSPCs as well. This leads to an increase in transgenically labeled cells that are budding from the dorsal aorta, shown here by live imaging. And we can show that quantitatively by FACS as well, where YODA1 leads to an increase in CD41 GFP low HSPCs, which you don't get if you do the experiment on a piezo1 mutant zebrafish background. This treatment with YODA1 can actually rescue the deficit you get in zebrafish embryos that have silent hard morpholino, so have no mechanical forces through their aorta at all. This can be somewhat rescued. And we think these effects are dependent on YAP because treating YAP homozygous mutants with YODA1 no longer leads to a statistically significant increase in HSPCs. And we think this really might be in part by YODA1 stimulating the expression of this battery of hematopoietic transcription factors that I had showed you earlier from our single cell RNA-seq data that we know are positively regulated by YAP. And finally, we think that this stretch piezo1 YAP axis might be conserved in vitro in our human cell culture system. This is immunohistochemistry for YAP staining in this iPS-derived CD34 culture, where YODA1 treatment leads to more YAP in the nucleus. And what we see is that we get an increase in the expression of hematopoietic transcription factors, and real notably here, canonical YAP target genes as well. So that leads me to my conclusions. What I've told you today is that we think that this cyclic stretch from blood flow helps drive EHT and HSPC formation in part by stimulating YAP activity. We think that molecularly, one of the things that YAP is doing is maintaining a self-renewal and cell cycling and metabolic signature in hemogenic endothelium to help maintain this differentiation state as cells are going through the process. We show now for the first time that TAZ, in addition to YAP, can regulate hematopoiesis and may do so by synergizing with RUNX1 on RUNX1 enhancers. And that we think one of the ways this might be being sensed is by activation of PH01 to get you to this YAP mechanotransduction cascade. And in the future, we have a few experiments lined up that are underway to try and assess what are some of the functions that might be specific to TAZ. We're willing to try and pinpoint YAP genomic occupancy with some of these genes that we think YAP regulates, as well as testing different YAP and TAZ variants to identify which DNA binding partners they work with to promote hematopoiesis. And finally, we'll be very interested to see if tweaking this axis pharmacologically in vitro might actually improve the engraftability and stem-like performance of HSPCs derived in vitro. And so with that, I come to my acknowledgements. I want to thank everybody on this slide, lots of collaborators, especially my supervisor, Tristan North, for being a great mentor, and a lot of people in my career who have helped me get here today. Some of them are on the call, and all of you for your attention. And I will end with a plug for myself that I'm looking for faculty positions this year. So I have a real interest in trying to understand these molecular mechanisms that underlie form and function integration. So if that sounds like something that your department might be interested in, you can find me on Twitter or by email. So I will go ahead and stop there, and I'd be happy to take any questions.
Thank you so much for a really wonderful talk, Wade. That was really gorgeous. While we're waiting for people to type in some more questions, I wanted to ask you a little bit more about the difference between YAP and TAZ. So you see this really strong upregulation of RUNX1 when you have TAZ, and do you have any hypotheses as to what is driving that in comparison to YAP?
Thanks. Yeah, that's a great question. So some of the data I didn't show is that we made a heat shock inducible constitutively active TAZ variant in the zebrafish that also increases RUNX1 expression. What we haven't picked apart yet is which promoter elements might be responsible for that. So is it YAP and or TAZ on TEAD enhancers, or might there be a role for something like a TAZ and RUNX1 interface on RUNX1's own enhancers? So it's known that RUNX1 can auto-regulate itself, and then there may be a way here for a mechanical cue to help augment that auto-regulation. So that's something that would be very cool, and I've kind of got some fish on the pipe that might help answer that question.
Fantastic. Joey Gersey would like to congratulate you on an amazing talk. And as you mentioned, this mechanism to help maintain HSPC state is restricted to HSPCs to differentiate into blood lineages. And so is this restriction true?
Hi, Joey. Thanks for the question. I'm just reading it again to make sure I got it. Yeah. So I think what it's doing at this stage of development is putting the brakes on differentiation into mature lineages so that the cells have more time to lean into the hematopoietic stem cell fate. So I don't know if that exactly answers your question. It's we know that YAP overexpression doesn't cause any problems with differentiation to the other lineages later in development. And we also know that if you lose YAP function in an adult mouse, for example, there are no problems in the hematopoietic stem cell compartment. So I do think that this is a developmental timing requirement that helps get this population created, and then it may be dispensable thereafter. Fantastic.
Peng Guang asks, so HSC PDCs are induced from the ventral side of the dorsal aorta, does differential mechanical force play a role in this induction?
Hi, Peng. Thank you for the question. That is a great idea, and I actually didn't draw attention to this, but if you make movies of the blood flow in this dorsal aorta in zebrafish, you really can actually see that the ventral wall is pulsing up and down with the blood flow, and then the top wall is like totally static because it's sort of slammed against the hypochord and it can't move. And so that's by way of saying that for sure there's probably differential mechanics, and I think that's a good open question is maybe how much of that leads to polarization, for example, in the genetic programs in the two sides. So that I think is not clear. I will say that if you take a silent heart morphin, for example, which has no blood flow at all, the RUNX1 expression that is left stays polarized to the bottom. So at least that aspect is maintained.
Fantastic. Sally Moody asks, are there any clues as to which members of the TEED family are involved?
Hi, Sally. Thank you. That's a great question. Short answer, no. At the moment, I don't have an idea, but I will go back and mine the transcriptomic data that we've generated and others have generated. We could at least take a start by saying which TEEDs are expressed in hemogenic endothelium. Knowing zebrafish, I know there's at least 4 TEED transcription factors. There's probably some redundancy baked in there. It's one of the reasons why dominant negative tools are a way to at least try and get at, is it a TEED, irrespective of which one? But that is something we can follow up with for sure.
I have another quick question about cyclic stretch. So you said that cyclic stress is really required for hematopoiesis maintenance. And I'm wondering if there's anything known about cyclic stress differentially affecting YAP or TAZ.
I don't know about differentially activating YAP or TAZ, but what I can tell you is that wall shear stress, for example, so this persistent laminar wall shear stress seems to ultimately kind of exclude YAP and TAZ from the nucleus. So people in the vascular world call that atheroprotective flow. It's not disturbed, but sort of cyclic flow or stretching, that is activating YAP and TAZ. And so I know that's not exactly what you asked. I don't know if I can pin down preferential activation of one or the other, but the two forces notably have different effects.
Fantastic. And one more question, also from Sally. Is it possible to use the PDZ domain to do a co-IP to determine binding partners during hematogenesis?
That is a great idea. It would definitely be possible. Potentially an experiment we could be doing in our human IPS hemogenic endothelial culture, because we have more material there to work with. That might actually be a really good way to do that, yeah.
Fantastic. Well, with that, I think I'd like to thank Wade for our really lovely talk and for everyone's attention and discussion. So, and to also thank you for our first speaker, Jennyfer, for a really excellent talk. So this seminar has been recorded and will be available on the SDB website next week. We invite all of you to join us next month on Friday, April 12th, when Iket Sonfo from Stowers Institute for Medical Research and Catherine Goodwin from MRC Laboratory of Molecular Biology will be presenting their work. Thank you all for coming.